Hello, this is part two of the presentation of this article in the February 2020 Science Advances Journal, and this is given as part of the Host Pathogen Interaction Journal Club at the University of Maryland. This video is preceded by an introduction and part one. A summary of part one of the presentation is presented here, so that if you wish to review, you can just stop the recording at this point. We now move on to the author's exploration of the erythrocytic blood stage of both the wild-type PBNK65 superscript S and its mutant form PBNK65F. Initially, they infected the mice with either the wild type or the mutant with 10 to the 6 parasites per mouse, and found that the survival rate, the parasitemias count, and the hemoglobin levels were similar, as you can see in the graphs. But the authors also collected sera on days 3, 7, 11, 15, and 19. As you can see, on day 7, the serum levels of the interferon gamma, the master enhancer of T helper cell 1 differentiation, as well as the levels of interleukin 1 beta and tumor necrosis factor alpha, cytokines that also enhance Th1 cell differentiation, were significantly higher in the mutant PBNK65 superscript F infected mice. Conversely, on day 7, the levels of pro-inflammatory regulators and chemoattractants, as shown here, were significantly higher in the wild-type infected mice. To further characterize the immune response to the wild-type and the mutant, spleens were collected on day 6, 10, 15, and 19 after infection. The total number of germinal cell B cells and B cells of the plasma cell lineage and the follicle helper T cells were quantified. The number of these type cells peaked similarly at day 10 and 15. However, on day 19, although all these cell types decreased in number from wild type and mutant infections, the numbers were significantly higher in the mice infected with the mutant PBNK65 superscript F. To determine if the increase in germinal center B cells and B cells of the plasma cell lineage and TFH cells in response to the mutant infections were dependent on the interferon gamma, a comparison was made by infecting wild type with interferon knockout mice. It was found that these types of cells indeed were interferon gamma dependent. Studies have shown that the architecture of the spleen is disrupted in the acute blood stage of malaria. A histopathological evaluation of spleens taken from wild type and the mutant have similar B cell follicles until 19 days after infection. At that point, the spleens of the wild type infected mice show disruption of the B cell follicle structure. noting a more persistent germinal center B cell and plasma cell lineage response to the wild-type infected versus mutant mice, the levels of total and infected red blood cell specific immunoglobulin M and immunoglobulin G at days 7 and 19 after infection. The sera obtained on days 7 and 19 after infection showed similar levels of total IgM. However, on day 19 after infection, the levels of total IgG were significantly higher in mutant infected mice. Infected red blood cells were lysed on day 7 and 19. On day 19, the specific infected red blood cell immunoglobulins IgG, Ig2b, and Ig2c were significantly higher, but IgG1 and IgG3 were not, indicating a predominant Th1 type immune response to mutant parasites. This is consistent with the earlier observation of a T helper cell 1 type response. Noting that the mutant PBNK65 superscript S did elicit an immune response late in the infection, the authors explored reducing the parasite burden to see if it would result in survival and protective immunity. They implemented the reduction by two methods. One was treating the mice with a non-sterilizing dose of chloroquine, now famous, and the other was by reducing the inocula. For the chloroquine method, they infected mice with both the wild type and the mutant, let the infection progress for 8 days. Then some of the mice were treated and some were left as controls. The controls all died by day 23. The treated mice were treated from day 8 to day 16 and then stopped treatment. By day 40, 80% of those infected with the wild type had died. In contrast, 60% of those infected with the mutant survived. In addition, they determined that the mice which survived also survived a following challenge infection with mutant and 60% protected against reinfection by wild type. The other approach reduced the inoculation to 10 to the second power. 70% mice infected mutants did not develop detectable levels of parasitemias and survived the infection. In contrast, none of the mice infected with the wild type survived. When this was repeated with interferon knockout mice, none of the mice survived. This supports the previous experiment that the interferon gamut is key for the immune response. Finally, to determine if the progression was dependent on adaptive immune response and not on intrinsic growth properties of parasites, wild-type mice and mice deficient with RAG2, recombination activating gene 2 protein, were infected with wild-type and mutant. Both progressed to high levels of parasitemias, and thus the low inoculation mutant infection was lost in the RAG2 knockout mice. In the discussion part of the paper, they believe that understanding, and rightfully so, the genetic factors of Plasmodium will likely contribute to developing a vaccine, hopefully in the future. The binding domain of the ApiAP2S is conserved in the human malaria parasite Plasmodium falciparum. So this would be an important study for developing the vaccine. Evidence of the SNP in the ApiAP2 DNA binding domain of the transcription factor is a virulence factor in the mouse parasite PBNK65 and differentially expresses 46 genes involved in the blood stage of the infection compared to the mutant. Mice were unable to elicit an effective immune response to the wild type even at low doses, while an effective immune response to the mutant was mounted and gave mice resistance to reinfection. The immune response to mutant PBNK65 superscript S was interferon gamma dependent. They believe that they're the first ones to report any API-AP2 family member in host pathogen interaction, and they believe that this is also the first characterization of a functional role for existing API-AP2. Now, just a couple of quick comments from myself. It seems to me that while the paper demonstrated that the SNP, the single nucleotide polymorphism in the transcription factor, is a virulence factor, it is one or combination of the 46 genes which are differentially expressed, which are, I believe, the ultimate virulence factors. As days 7 and 19 seem to be pivotal moments in the response to the wild type and mutant, it would be interesting to do RNA-seq analysis at those time points. This concludes my presentation. I am certainly interested in your feedback and hope everyone is in good health.